Now, the second presentation tonight is from Emmeline Hill. Emmeline, of course, comes from a preeminent family in the world of racing, but she's an Associate Professor of Equine Science at UCD, and she was the co-founder of Equinome Limited about 6 years ago. She's Chief Science Officer of Plusvital, and she has been to the forefront of thoroughbred performance genomics during the last decade, and she has published more scientific papers on equine exercise genomics than any other researcher worldwide. In 2010, the publication that described the discovery of the speed gene in racehorses coincided with the launch of Equinome and the company's first commercial genetic test. And in UCD, she leads a nearly €2 million funded research program in equine exercise genomics, which is the largest of its kind in the world. Scientific American in 2012 wrote that when it comes to published academic research, Hill may be champion among the geneticists engaged in the search for the secrets of equine athletic potential. So here to unveil those secrets to you and reveal them to us all, please give a warm welcome to Emmeline Hill.
Thanks, Leo, and thank you very much for coming to be here this evening. And thanks to Lisa for her introduction to how genetics can be used in managing health. I think that's a very important place to start, because the goal of the horse genome sequencing project was specifically to improve the health and the welfare of the horse. And that is a goal of everybody who works in this field. We are living in a new era of breeding. It seems that now we have actually got to the point where people are using information, genetic information, based on modern scientific technologies to help to inform breeding decisions. And therefore, this is a new phase and a change in the way people have practiced breeding for possibly hundreds of years. So I'm going to tell you this evening about the work that has been done and the work that we've been doing to understand the genetic contributions to athletic performance and traits that are relevant to the thoroughbred. We launched a company, Equinome, that merged with Plusvital last year in 2010 just following the discovery that we were able to identify a gene that we called the speed gene, and using the genetic information, make a prediction about how a horse would perform on the race course. And this was the first time that this had ever been done. Since then, we have tested over 14,000 horses in our labs for the speed gene and our other genetic tests. And we have clients in all of the major bloodstock regions of the world in over 20 countries. One of the founding, I suppose, principles, or the core principles of the company is to do real science. And in order to achieve that, that means two things. One is to publish our scientific findings so that the data is peer-reviewed, and that it's not just us saying, trust me, I'm a scientist, that real science is scrutinized, critically scrutinized by peers and experts in the field. And we do our best to publish as much as we can. And as well as that, we have engaged some leaders in the field of equine genomics, and have a great team of researchers that have joined us in the last couple of years. And these people are experts in the area of mathematics and statistics, in veterinary science, in equine science, in computer programming and bioinformatics. And we engage and consult very closely with experts in the university, Lisa Katz being one, and others, leaders in the field, including Professor David McHugh, who's very much involved in cattle genomics, which is an area of genetics in which Ireland, in fact, are leaders. So there are a number of genetic tests that are now known, or there are tests for performance. Lisa gave a very good introduction into genetic tests for health traits. Many of those genetic tests are actually simple traits that exist in other horse breeds and not in the thoroughbred. Genes that have been identified in the thoroughbred to affect performance are the Speed gene, which I'm going to tell you more about. In non-thoroughbreds, in horses, in horse breeds that can perform alternative gaits, a gene that has been named the Gatekeeper gene has been identified by a research group in Sweden. This is the pacing gene, and this is very high frequency, or found at very high frequency in breeds that, as I said, that can pace or can perform alternative gaits. So the Icelandic horse that can perform a pace called the tolt, for instance, has this gene. There are genetic markers that have been identified for racing performance or elite racing performance. I'm going to tell you about some of the genetics that we have found underpinning this trait, but there are other companies and other researchers around the world that have identified genetic markers for elite racing performance. These have not yet been published, or they're certainly not in the public domain because of the, I suppose, commercial sensitivities around them. But we also know that there are published articles that relate to genetic markers for RLN. Lisa mentioned RLN earlier on. For fracture risk, and these are in thoroughbreds as well. For tractability, or the ability to train a horse, so the trainability of a horse. And in non-thoroughbreds, in the sport horse, for style and ability in free jumping, and a breeding value for competitive jumping. These haven't yet been translated into genetic tests, but there certainly are known genetic markers underpinning these traits. The Speed gene has, we discovered the Speed gene in 2009. And as I said, we have 14,000 horses that have been tested from belonging to clients all over the world. Earlier this year, in April, the end of April, Galileo Gold was tested for his Speed gene in order for his trainer and the managers of the horse to make the decision, to help them make a decision whether that horse would go to the Derby or not. He didn't run in the Derby. He was kept at a distance of a mile, which is the distance that best suits him, and I'm going to tell you about that in a little while. This wasn't the first horse in which decisions like this had been made, but it was the first public example of how this information is now being used within the industry. And I'm going to tell you a bit more about the actual information that we're generating and that we're providing to our clients and how it can best be used. First of all, what is genetics? So genetics is the branch of science that allows us to look at factors that are inherited from one generation to the next, and allows us to understand the differences between two individuals. The term genome is a term that's been used for the last 70 or 80 years to refer to the complete complement of genetic information that is the blueprint for an individual. Now, believe it or not, everybody understands genetics. Everybody in the thoroughbred industry understands genetics, because anytime anybody looks at a pedigree or evaluates a pedigree, what they're trying to do is to best guess which genes have been passed down from ancestors in that pedigree to the individual that's represented in that pedigree. So for hundreds of years, breeders have been using genetics. What we're doing now is that we're applying modern genomic technologies or modern scientific technologies to try to do something that people have been doing already for hundreds of years. The reason that we can do this is because in 2007, so it's almost 10 years ago now, the horse genome was sequenced. And this was a collaborative effort that was funded by the National Human Genome Research Institute, and a collaborative effort of 70 or 80 researchers from around the world that all came together with the single goal to sequence the horse genome for the improved health and welfare of the horse. So what is a genome? every living organism, you or a horse, is made up of billions, trillions of cells. And inside every single cell, housed in the nucleus, which is this part of the cell here, in every single cell in the nucleus is the genome, or the genetic material. The nucleus is the house for the genetic material, which in the horse is packaged into 32 pairs of chromosomes, one of each pair coming from the sire and one coming from the dam. The horse genome sequence is made up of 3 billion letters of the genetic code, and it's our job to try to understand what these letters are doing in the horse, and specifically what differences or changes or mutations in these letters, what effect that will have on the phenotype or on the physical characteristic of the horse. So if you imagine this blue bar is a chromosome. So the blue bar represents a chromosome, and the letters A, T, G, and C are the DNA running along that chromosome. So DNA is represented by the letters G, A, T, and C, and various different combinations of those letters spell out the genetic code for a gene, which is represented, genes are represented here in the orange blocks, to make a particular protein in the cell. There are 25,000 genes in the genome, and they're distributed along the chromosomes. Some genes are big, some genes are small, some are close by one another, and some are quite far apart. But in every instance, a gene has a unique sequence of these letters, and that unique sequence tells the cell to make a particular protein. Lisa mentioned simple traits and complex traits, and it's important, very important to understand this, that a simple trait is a trait that is influenced or caused by a mutation in a single gene. A complex trait, which is more common when we think about performance-related traits, a complex trait is one that is influenced by more than one gene, 2 or more genes, and also that has may also be influenced by the environment. So a complex genetic trait is one that where there are many genes involved contributing to the trait, and the environment may also play a part. There are 3 billion units of DNA, so 12 billion letters in the DNA code of the horse. We can now at any one time evaluate 670,000 units of information of these letters. Those 670,000 units of information essentially are representative of the whole. They represent bits of the DNA. So in Plusvital's genetic tests, we now use almost 50,000 genetic markers to inform our tests. And importantly, we have a very large sample base, or a research set of horses on which we can phenotype, as Lisa referred to, on the basis of their past racing performance. And that's the research set that we use. And we have started to develop, we have developed novel and very sophisticated statistical modeling, which is very similar to the approaches that are now being used in the cattle breeding industries to capture all of this information, bring it all together, and use this information to make predictions about risk for certain, whether it's risk for certain health traits or chances of performing at a particular level. So this has come a very, very long way in the last 6 years. From 7 or 8 years ago, only being able to look at a single gene at a time or a single genetic marker at a time, we can now look at all 25,000 genes at a time and 670,000 genetic markers. So you can see that the pace of the change, the rate of change of these technologies is accelerating extremely fast. So in order to understand the tests, I think it's a good idea to understand how we actually identify the genetic markers for them. First of all, we have to characterize the trait and understand the phenotype, which is Lisa was referring to in her talk. And we have to separate out the population into horses that can be characterized as cases or horses that are characterized as controls. When we have clearly defined the phenotype, then we can extract DNA from the samples, from tissue samples from that horse, whether it's blood samples or hair samples. And from that DNA, then look at the 670,000 genetic markers for each of those horses and compare the frequency of the letters or the letter differences between the cases and the controls. So let's say we have a particular genetic marker, one of these 670,000 genetic markers at a particular position in the genome can either be a letter C or a letter A. Now remember, you have two copies of every gene, one that comes from the sire and one that comes from the dam. So let's imagine that the C came from the sire and the A came from the dam. It could be the other way around, okay, in this instance. And what we find when we, in this example, in the cases, the C allele or the C type of the gene is more common among the cases than it is among the controls. And that's significant at the 0.05 level. So this suggests then that this difference in the genetic code may either be in a gene or close to a gene that is contributing to that trait, whatever the disease is or the trait that we're looking at. That's the premise behind all of this. But instead of looking at it one at a time, instead of looking at these C's and the A's and the differences, the frequencies of these letters one at a time, we can now do this in a highly paralleled manner by using technology known as a SNP chip. And what these SNP chips do is they allow us to put DNA onto them and get the answer, the C, A, G, or T answer for 670,000 units of information at a single time. So what we do is we run 670,000 simultaneous statistical tests to see if these letters are associated with the trait. Now, that's hard to imagine. 670,000 is a huge number. How do you sort of compute it all in your head? Well, what we do is we generate these plots, which are known as Manhattan plots. Each dot represents one of these 670,000 bits of DNA, and anything above the red line represents a genetic marker that is significantly associated with the trait. And you can see here that these dots, or these genetic markers, are all significantly associated with the trait. These are the ones that are most likely the most important genetic markers. But we're missing a huge amount of information if we only look at those small number that pass the threshold of what is known as statistical significance. These are informative in that they may be close to or within genes that are contributing to the trait, but we're missing an awful lot of information down here. So what we can now do, and the statistical approaches that we're now taking, is instead of just looking at the top genes, the very small number of genes that pass that threshold, we can now capture all of the genetic information that there is for a horse and put it together by using the sophisticated machine learning and predictive modeling, by putting all of this information together and making a prediction from all of the information in the genome. Using this approach, Plusvital has developed a range of genetic tests that are relevant for performance, and I'm going to be going into more detail about those in a few minutes. What's important about these tests, complex genetic traits, is that, as I mentioned, they're influenced by a very large number of genes, but also the environment. So what we have to understand in all of these cases is how much is it due to genetics and how much is it due to environment. And this is the term heritability that Lisa was referring to. We can only expect that there might be a genetic contribution to a trait if the heritability, if there's a level of a heritability, that it is actually passed down from one generation to the next. And so this is something that we have to understand for all of the traits that we look at. But before I go on and talk about the complex traits, I want to tell you about a simple trait, something that remarkably has upheld itself in the claims that we made 7 years ago, that there is a single gene that has a major contribution to a horse's performance. we didn't ever expect it as scientists. The breeding community didn't ever expect that there would be a single gene that would contribute to performance. But the more research we do, the more evidence there is that this is the case, and not just the research that we do, but researchers all over the world. So the original claims that we made, that there's a single gene that we refer to as the speed gene that contributes to a performance trait, has been entirely upheld. The reason we first started looking at this gene was because we didn't have the opportunity to look at 670,000 units of the genome at a time. This was before the horse genome was sequenced. We were only able to look at one gene at a time. So we had to take a scholarly guess as to which genes might be contributing to performance traits. And I had read a number of articles that a gene called myostatin contributed to unusual muscling in a range of species. This is a Belgian blue bull that has a trait called the double muscle trait. He looks like that because he has inherited mutations in his myostatin gene from both his dam and from his sire. So he has 2 copies of mutation in his myostatin gene. The dog in the middle is a whippet, believe it or not. This is known as a bully whippet. And that dog looks like that because he also has 2 copies of a mutation in his myostatin gene, one inherited from his mother and one from his father. Now, dogs that have 1 copy of the mutation are better racing dogs than these dogs. These dogs actually aren't good racing dogs because they have too much muscle bulk. But dogs that have 1 copy of the mutation are actually better racing than dogs, better racers than dogs that have no copy of the mutation. And the mouse on the right is known as the mighty mouse. He actually has no myostatin at all, and he has because he has no myostatin, his muscle has bulked up. Myostatin acts as a negative regulator of muscle mass, so it acts as a brake. The more you have, the harder the brake is on. As you have less myostatin, you remove the brake, and muscle can bulk up. So we started looking at the difference in the DNA of the myostatin gene in the thoroughbred because nobody had ever published on it. Nobody had looked at it. We didn't know if there were any mutations in the myostatin gene in the thoroughbred. And when we read the sequence of the letters of the DNA code of the myostatin gene in thoroughbreds, we found that at one particular position in the gene, there could be a letter T or there could be a letter C. All the other letters are the same, except for this T to C change. There are a few other differences, but this is the one that we focused on. Now, of course, as I said, all individuals have 2 copies of each gene, one inherited from the sire and one inherited from the dam. So if an individual inherits the C-type chromosome from the sire and the C-type chromosome from the dam, then their genetic type is known as CC. This is the genotype. If they inherit a C from the sire and a T from the dam, or the other way around, a C from the dam and a T from the sire, their genotype is CT. And if they inherit a T from both parents, then their genotype is TT. So there are 3 possible combinations, CC, CT, or TT. And every single thoroughbred and every single horse in the world must be one of those 3 types. What we did then is that we separated out the horses into whether they were sprinters or whether they were won their best race over longer distance racing. And we found that horses with the CC genotype were best suited to short distance races. CTs were the middle distance type horses, and TTs were horses that preferred distances that required more stamina. And this was a highly statistically significant relationship between this genetic marker and the trait of interest, which is the distance of the highest grade of race that a horse had won at. So it's highly predictive of the best race distance for a horse. You can see here's the Manhattan plot for this particular trait, and you can see that so much of it is, so many of these dots are above the line that there's very little else here showing up below the line, which indicates that there is almost entirely a singular influence on the distance trait from this particular gene. This is the distance range in Europe for CC horses. And of course, in different regions, for instance, in Australia or in the US, where there are different opportunities or different distances in the different regions, therefore there are different opportunities to race. The CCs will have a slightly different range, the CTs will have a slightly different range, and TTs and so on. So this is the distance range for CCs that we have determined in Europe. The majority of them, almost all of them, have won their best race over distances up to a mile. And very, very few of them have won at the elite level, at group race level, have won their best race over distances greater than a mile. The CTs are more versatile in terms of their distances. These are the type of horses that can perform well over the shorter distances as 2-year-olds and train on well over the longer distances as 3-year-olds as they get the opportunities. And the TTs, remarkably, are at the other end of the distance spectrum. Over 80% of TTs have won their best race over 10 furlongs and above. So it's in very stark contrast between the TT distances and the CC distances. In Europe, there's quite a balanced population in terms of the speed gene types. About 25% of the population are CCs, 50% are CTs, and 25% are TTs. This distribution is very different in different regions of the world. In Australia, for instance, about 45% of the population are CCs, 45% are CTs, and less than 10% are TTs in Australia. This is because of the race pattern that's set in Australia is very much targeted towards shorter distance, early 2-year-old racing, in which CC and CT type horses excel and TTs struggle. Now, many people may counter that this information can be determined from pedigree. All you have to do is look at the pedigree and you can see on the dam's line is full of stamina. So why do we need a genetic test? Or the stallion side, the sire side is full of speed. So of course it must be a sprinter. Well, if you have a CT mare and you take a CT mare to a CT sire, cast your minds back to insert science, Leo. You might remember Mendel's peas and these Punnett squares. And I always call this Punnett square Mendel's peas in real life. If the dam passes on her C-type gene to the foal and the sire passes his C, then that foal is a CC. If the dam passes her C and the sire the T, then it's a CT foal. Another CT foal, and if they both pass on their T, then it's a TT foal. So in this particular instance, if you take that mare to the stallion, same stallion 4 times, those foals will have identical pedigree pages. They'll have identical pedigree pages, yet genetically they're very different. And what this means is this translates into being very different horses on the race course, in training and on the race course. So the utility of this is that you can make predictions for your matings, how you can use this information. You can use the information to more often produce the type of foal that you desire. And you cannot always predict the outcome from pedigree. This is just an example of a set of horses that are a son and a grandson of probably the best or one of the best stallions in the world, who was a TT. And he had a son who's a CT, who had a son who was a CC. But there have been often questions as to why progeny of a particular stallion or progeny of a particular mare may actually perform differently. It's because of the combination of the genetic material that has been passed on from both parents. So this can now and is being used to inform breeding decisions. Now, breeding, I suppose, is a genetic lottery. You never know what's going to come together. But what you can do is you can use the information to load your dice, so to speak. If you know the genetic information for the mare, and you may know it for the sire, then you can use the information to more consistently produce the type of foal that you want. So if you want TT horses that are best suited to Derby distances, for instance, then if you have a TT mare, then you can send her to a TT stallion and you will be absolutely guaranteed to produce a TT foal. And similarly, if you have a CC mare and you want to ensure that you're producing sprint types, then you can send that mare to a CC stallion. In practical terms, as I said, this is being used to inform decisions within the industry. This is an example of a set of 142 horses that were in training with the same trainer. These are Jim Bolger's horses in 2007 and 2008. He didn't know the genetic types of his horses at the time. That was because we hadn't discovered it at that point. So he wasn't training these horses according to his knowledge of the genetic information. But what we found when we retrospectively assessed the performance of these horses as 2-year-olds, we found that the CCs and the CTs on average returned their training fees in earnings and the TTs did not. And this is because 2-year-old racing is restricted to the shorter distances and the TTs aren't suited to that. And also because of the rate of the maturity of the musculature of the TTs is slower than the rate of the muscle development of the CCs and the CTs. And this translates directly into performance as 2-year-olds. Now, we were slightly concerned that maybe there was some bias in there, that there were some stallions maybe that were better stallions that were producing the CC and the CT types. So we looked at the performance of progeny of a single stallion that were all trained in that same trainer's yard. And all of the foals, or all of the progeny that were racing, there were CTs and TTs. This tells us that that stallion must have been a TT. And when we looked at the performance of these horses, it was strikingly different. 73,000 on average in earnings versus 3,000 on average in earnings. Yet these horses were all by the same stallion, were all given exactly the same opportunity in the same trainer's yard. Jim happens to be a breeder as well, and he recognized that if he produced fewer TTs, that he'd get a greater return on his investments. So with the knowledge in 2010, he started making his breeding decisions using this information for his mares and for his stallions. And within a single breeding season, he reduced the proportion of TT foals in his foal crop from 16% to 5%. How this actually translates is that in recent years, he's had a considerable increase in his strike rates for his 2-year-old crops and increase in earnings. So his 2-year-old strike rate went from 9% to 18% in the space of 3 years, and earnings per run more than doubled for his 2-year-old horses. What's interesting is that when we looked at a set of 218 horses that were not trained by Jim, and we don't know if these particular trainers knew the genetic type of their horses or not, but when we retrospectively looked at when CCs, CTs and TTs had their first race, we found that TTs won their first race on average 8 months later than CC horses, but there was no significant difference in the age that these horses had their first start. And this seemed to indicate to us that the trainers weren't able to figure out which horses were the CC types and which horses were the TT types. The reason that this translates as it does on the race course is that CC horses have 7% more muscle mass as 2-year-olds than TT horses. And CC horses also have a greater proportion of the type 2B muscle fiber types, which are the fast twitch fiber types, than TT horses. We know this because we've looked at a set of Japanese horses that were all kept in the same, kept and raised in the same training facility, and their body composition was measured when they first came into the yard and then during their first year of training. And it was found that the CC horses had significantly more muscle mass than TT horses. Well, you might say, well, we can see that anyway. You can see, a CC is more muscular, and you can just look at its conformation and you'll know that it's a CC. In many cases, that might be the case, but it depends when you want to start making your decisions. It depends on when you want to use the information. The interesting thing is that if you extrapolate this back, this is when these horses are yearlings. If you extrapolate these lines back here to when they're foals, there is no way that it can be predicted, the genetic type of those foals, from looking at their body composition when they're born. So the whole point of this is not prospective or retrospective, because hindsight is wonderful. It's about being able to use the information to make predictions about the future and better management in the future for these horses. So that's the speed gene, and as I said, it's remarkable that still 8 years later, there is no contest amongst the scientific community that this is the case, that this gene has such a major singular influence on the distance trait. But using our new, more sophisticated technologies that can capture information across the entire genome, we've been able to capture subtleties that we weren't able to identify before by looking just at a single gene. What we've been able to do is to separate out the CCs into horses that are short CCs and long CCs, so fit into the lower end of the CC spectrum and the upper end of the distance spectrum for CCs. Similarly for CTs, short CTs and long CTs, and short TTs and long TTs. We were able to develop this because we had information for over 2,000 horses in all of the major race regions. Almost 1,000 of these were group race winners. And as I said, because of the differences in the racing opportunities in the different regions, we've tailored this particular test for the major racing regions globally. So this is the Manhattan plot that I showed you earlier, showing that there is this major singular influence of the myostatin gene contributing to distance. But what we hadn't been able to capture until now was this more subtle information here that is also seems also to be contributing to this trait. Now, all of these other genetic markers that we can see here are likely modifying in some way the trait and are contributing in a small but in an additive way to distance. So this is an example of how we've been able to separate out these distances. This is in Europe, the average best race distance for a set of almost 400 group level performers into CC short, CC long, CT short and long, and TT short and long. So this is just enabling us to get much greater resolution on our first test, on our speed gene test, and to provide a greater level of detail and level of information for our clients. And this is how all of our tests are going to evolve. The more information that we can gather, the greater the research set that we have, the more information that we have, the more fine-tuned we are going to, the more fine-tuned are the tests going to be. This is the same for our elite performance test. You can see here that we're now on elite performance test version 3. We first launched an elite performance test in 2011, where we captured information for 80 genetic markers. Our Elite Performance Test version 2 captured information from 1,000 genetic markers. We're now capturing information from 50,000 genetic markers that are representative of the 670,000. Our next version will capture even more, and the next version, and so on, until we get to the point where we will be capturing information from the entire genome sequence. So this is the evolution of how these tests are developing. Our Elite Performance Test provides 3 bits of information. We provide a racing value, an inbreeding value, and a breeding value. And each of these provide different information provide information that can be used differently according to our clients' needs, but is all based on information from these 50,000 genetic markers. The racing value is probably the one of most interest, and that's what I'm going to provide a little bit more detail on. We captured the information for the 50,000 genetic markers, and on the basis of separating out horses into, very crudely, good or bad horses, so we have our set of group race winners, and we have a set of horses that have never won races, and then everything else in between as well. We separate out our population, clearly define the phenotype, which is what we were talking about earlier on. So when we have all these knowns, we can profile the genetic information at 50,000 units of information for all of those known horses. That's our research set of horses. So that when we take a blood sample from an unknown horse, we can say how closely does it match the elites, and how closely, or conversely, how closely does it match the poor performers. So from this information, we assign a score of 1 to 4. 1 being the highest chance of being an elite performer, 2 the next highest chance, 3, and then 4 the lowest chance of being an elite performer. We've actually just added on for this new test a score of what we call 1 star. This highlights the class 1 horses that are at the very, very tippy top of the range. So this is a very large sample set now of 4,000 horses, which is our reference population. And almost 1,000 of these have won at group level. The other thing that we've done is we've included horses that have raced in all of the major race regions. So this is not just a Europe specific test. In the general population, let's say we took a set of 100 random horses, thoroughbreds, in the general population, we would expect 20 of them to be class 1, 30 to be class 2, 30 to be class 3, and 20 to be class 4. So that's the percentage that you would expect in a general random population. What we see when we look at a set of group winners or group performers, that this distribution is very different. There's a much higher proportion of class 1 stars and 1s in the elite population than there is class 4s. The class 1 stars have, on average, a 10% chance of being black type race performers, which is twice that expected in the general population, which is at about 5%. And this is a figure that's come from Weatherby's, where we expect about 5% of the population to be group race performers. So this allows us, on a population level, to identify the sets of horses that have the greatest chance of racing success. So in our test development, we use horses that have already raced in order for us to assign them into the categories of whether they're group performers or poor performers. But the real, I suppose, validation of these tests is looking at horses then that were not in the validation or were not in the research set that have raced. And we've started to evaluate this in a range of different farms around the world, and found consistently that the higher performing horses, there's a greater proportion of class 1s versus class 4s. So this is a case study of 2 leading UK farms where the class 1 horses were 4 times more likely to be black type than class 4 horses. The class 4 horses were 2.5 times less likely to be black type than average across the farms. And the class 1s, 2s, and 3 horses had a 1.7, or let's say a 2 to 5 times higher win to start strike ratio than the class 4s. So in all of these parameters of racing success, for these horses that were not used in the research set, it's panning out that the higher performing horses have the higher genetic score. We also provide to our clients a breeding value. The breeding value is calculated in a similar way to the calculation for the racing value, but it's slightly different in that the breeding value is not based on the genetic information that that horse has. It's based on the potential for that information to be passed on to the next generation. So there aren't interactive effects between the dominance and recessive, which is something that Lisa mentioned, because you're not going to pass on dominance and recessiveness. You're passing on just one copy of the genome. So what we're looking at is the chance of passing on a favorable set of genetic information or genetic markers to the next generation. And looking at our breeding value, we've found that horses that are class 1 on their genomic breeding value have produced twice as many stakes winners as class 4 horses. That's 11% versus 6%. So that's a huge difference in the production of stakes performers from a class 1, from a horse with a class 1 breeding value. A new test that we've developed just this year, hence it's version 1, is the chance of a horse having a race course start. And I was absolutely shocked when I heard the statistic. The number of thoroughbred foals that don't ever make it to the race course is in the order of 30 to 40%. And some people have even suggested that it may even be higher. So what's the reason for this? Why are horses not making it to the race course? And can we understand from a genetic perspective what this is? And with this information, can we manage these horses better in order to give them a better chance of having a race course start? So what we did is we developed a test that predicts the chance of a horse having at least one race course start as a 2-year-old or a 3-year-old. And we ranked them genetically as either being genetically higher potential for a race course start as a 2-year-old or a 3-year-old, medium potential or lower potential. And in this particular case, the population is equally divided amongst the three. So 33% of the entire population are higher potential, 33% medium potential, and 33% lower potential. Over 4,000 horses were used in the development of this test, and 14% of the horses were unraced. What we found is that when we looked at horses that were genetically higher potential, the higher potential colts had a 1.6 times greater chance of having a race course start than the lower potential. That translates, in other words, they had a 60% greater chance of having a race course start than horses that were genetically lower potential. And I think that's a huge, huge difference. What we don't quite understand yet is what are the genes that are contributing to this lower potential? It may be that these horses are more prone to injury. It may be that these horses have an underlying poor performance trait that we haven't matched up quite yet. It may be that they're just not suited to the environment for racing and training. They just don't like it. There may be a behavioral aspect to it. Or it may be that actually maybe they're just no good and they've been sent home. We don't know the reason for this. But what's striking is that even though we don't know the reason for it, we can identify the genetics for it. It's not as strong for the fillies, but still, the lower potential fillies have a 20% lesser chance of having a race course start than the higher potential. As I said, that's in a research environment. What about in a real life environment? In Australia, we tested 420 yearlings at the sales in 2014. They have all now completed their 3-year-old season. 2016 was their end of their 3-year-old seasons. They all had a chance to have at least one race course start as a 2-year-old or a 3-year-old. And these horses were tested. We took their DNA when they were yearlings, so we had no idea how they were going to perform on the race course. The results were remarkable. There was a higher run rate. There was higher earnings. There was a higher strike rate, a higher win rate for the higher potential horses than the lower potential horses. But there was no difference in the sales price. So the buyers were unable to distinguish between the higher potential, the medium potential, and the lower potential. What I didn't mention is that the higher potential horses had statistically significantly higher percentage of horses that had raced than the lower potential cohort. So if they were genetically a higher potential horse, they had a significantly higher chance of having a 2-year-old or 3-year-old race course start. They earned more, they ran more often, and they had a greater strike rate. And you can see here the difference. This is the percentage of horses that were unraced among the higher potential and the percentage of horses that were unraced among the lower potential. Almost twice as many. And for all of the parameters of racing success, this translated directly on the race course. So I think this is absolutely remarkable. Even though we don't know the genes that are underlying this trait, we're able to identify genetic differences. Finally, it has been suggested that there may be genetic differences that are contributing to a horse being able to race preferably on either dirt surfaces or turf surfaces. And this is obviously particularly relevant in North America, where there's dirt and turf racing. So this is a test that we've developed for our North American clients, where 264 horses were used in the development of the test. Horses that had won or were placed on one or other surface, but not on both. So we were able to distinguish between horses that were really dirt horses and horses that were turf horses. And we're able to very clearly make predictions about horses that are best suited to dirt and horses that are best suited to turf. Now, it's been recognized that there are sires that are better dirt sires, or there are sires that are better turf sires. That suggests that there is an inherent genetic contribution to this trait. Again, we don't know particularly why it is, whether that's whether it's a conformation trait, whether it's genes that are contributing to different conformation of the hoof or different conformation of the individual, whether it's metabolic genes that are different between the dirt horses and the turf horses. Again, we haven't got to that point yet, but I think it'll be very interesting to see what exactly the genes are that are contributing to these traits. But irrespective of knowing what the actual genes are, we can make very strong predictions as to how horses are likely to perform by taking a blood sample, taking the DNA from that blood sample, running it through on our 670,000 chip, running the genetic analysis through it, and determining whether that combination of genetic information makes that horse a turf pro, which means that horse has an 80% chance of being a turf horse, or whether that horse is a dirt pro, which means that horse has a 76% chance of being a dirt horse. So this information can be used in a variety of ways, and I've touched on quite a few different ways that the information can be used and really, as Lisa really was making the point, that this information, in the same way as the single gene tests, in the same way as any other information, it's information that will help to empower breeders, owners and trainers to make better informed decisions about their horses' care and management. And that's really the underlying point of doing all of this. So foals and yearlings can be used to test at a very early stage the potential for a particular trait or the risk for a particular trait. And therefore you can manage those young horses in a particular way. For horses in training, to reduce operating costs, to optimize training and racing strategy. And we've seen this great example of Galileo Gold earlier this year. We have many stallion managers around the world that are now advertising the genetic types of their stallions to promote their stallions and to attract the mares that are most suited to those stallions on the basis of the genetic information. And we're delighted to see so many people making that information available to breeders. And of course, for broodmare owners and for breeders to make decisions about their broodmare band, to optimize their the horses that they're breeding from, and to make more informed decisions for each and every mare to get the best and to maximize the genetic potential of that individual. So we firmly believe that genomics offers a better future not only to the individual horses, but to the industry as a whole. Not only to enable horses to race at their ideal distance or the ideal surface, to ensure more foals make it to the racetrack. In terms of the population as a whole, that is going to hopefully, if it were applied, to be a population-wide improvement in the health of the population. To breed broodmares to their strengths and to avoid their weaknesses, so that you're maximizing the genetic information from that mare being passed on to the next generation. And to use the information to reduce the incidence of negative health traits in the population. All of these have got to be positives for the industry, and most importantly, positives for the thoroughbred horse that we all love, and which is why we're all here. I've mentioned a number of traits. Lisa's mentioned a number of health traits. And there are some things that we're working on, and there are some traits that other people around the world that are working on. But most people in the industry will recognize heritable traits because, you know, like a lot of these things, old wives' tales often have a real scientific basis to them. And what we're trying to do is to use science to try to understand the basis for those suggestions. So if there are suggestions that particular stallions pass on certain traits, it may be that there's a genetic contribution to that trait, and that's what we're looking at. So some of the more common health traits like fracture risk, roaring or RLN, bleeding, tying up, and OCDs, all of these have been indicated to have a heritable component, and it's very likely that in the not too distant future there will be predictive tests for some, many, or perhaps even all of these traits to help us to better manage our horses. The reason that we can do this now is twofold. One is in collaboration with vets like Lisa, people in the industry that have helped us to characterize the phenotype of the horses. But not only that, the technologies have evolved and have developed at such a very rapid pace in the last 5 to 10 years that we can now capture a huge amount of genetic information. And we can use sophisticated modeling, statistical and mathematical modeling, and apply this to this problem. And we're very fortunate to work with some of the best people in the world in this area. So the limitations to advances in genetic technologies, or advances to genetic information in the thoroughbred industry, is not anymore the technologies. For such a long time, the thoroughbred industry the horse industry lagged behind other species in terms of the genetic tools that we had. We don't have that problem anymore. We have these tools. We can now use these tools to the benefit of all. These technologies have already been demonstrated to be a huge success in other production animal industries. For instance, the cattle breeding industry. In the last 60 years, the average milk produced from a dairy cow has gone from 5,000 pounds of milk in its lifetime to 21,000 pounds. That's a huge, huge advance using genetic technologies. Imagine the improvements in the health and the welfare of the thoroughbred population if we were to use this information in the same way. Thank you very much.